Good afternoon, everyone, and thank you for joining us. Welcome to the latest session in DHA Laboratories' educational webinar series. We're glad you carved out the time, and I think you're going to walk away with something you can use in practice right away. Today's topic is one that comes up constantly in clinical work, but rarely gets addressed at the root. Autism, ADHD, and anxiety are all usually treated as separate conditions with separate symptoms. What often gets missed is how much biochemistry they share underneath. Oxidative stress, methylation, mitochondrial function, nutrient status. These are the systems driving so much of what we see, and they're measurable. That's the whole premise of today's session. How to use lab testing to find the real drivers, prioritize what to address first, and personalize nutrition in a way that actually changes outcomes. Introduction to many of you. Julie is a certified nutrition consultant and one of the most respected voices in the country on nutrition and biochemistry for complex neurological and developmental conditions. She's spent her career turning complicated science into something clinicians can act on. On the personal side of things, I've had the privilege of working with Julie for more than 5 years, and beyond being a brilliant practitioner, she's someone I'm proud to call a friend and a colleague. I assure you, you're in very good hands today. We'll have a live Q&A with Julie and the DHA team right after this presentation, so stick around, keep your questions coming as we go, and Julie, thank you for being here. The floor is yours.
Hi, Dan. Thank you so much. I'm very happy to be here for this. I'll just go ahead and jump in. Let me share my screen. As Dan mentioned, today we're going to talk about autism, ADHD, and anxiety, as well as other related conditions, really diving into the biochemical patterns and using laboratory markers to guide targeted nutrition. So I am a certified nutrition consultant. As Dan mentioned, I have a master's degree in medical nutrition. I have 25 years of experience working with autism nutrition and children and families. I lecture all over the country and actually a little bit throughout the world. I work with both lecturing to parents as well as clinicians. I teach practitioners in my bio-individual nutrition training, and I work with families with my organization Nourishing Hope. And so, and I published 2 papers. So in the Journal of Nutrients and the Journal of Personalized Medicine, 2 papers on the effectiveness of diet and nutrition intervention for autism specifically. And I wrote a book, The Personalized Autism Nutrition Plan. So I'm really excited to jump into this today to talk about autism, but I actually started with my learning in ADHD, and the reason I ended up in autism was because they have so many similar foundational underlying factors and nutritional approaches that can help. And so very early on, I learned that autism was something that could be improved by using a personalized diet and nutrition approach. And so that is actually what I ended up spending most of my career doing. And so I'm really excited to share that with you today. So neurological and neurodevelopmental conditions, they are increasing, and symptoms are what often get treated or what get the attention, but often the underlying biochemistry is overlooked as a factor. And so they share similar underlying biochemical patterns. And so using laboratory testing can really help guide a personalized nutrition approach that can be very, very helpful. And when we see these similar underlying factors, we often see that even people with autism often have comorbid conditions of ADHD and autism, but also people could have just one of those individual diagnoses, but have that similar underlying factors as a framework or as contributors. So these conditions are heterogeneous, which means that there's no single cause or one single nutrition approach. So they all present differently because they have different underlying factors. And some of the key ones are oxidative stress, methylation, imbalances, mitochondrial dysfunction, nutrient deficiencies or nutrient imbalances like zinc or copper or folate, B12, B6. Those are just some of them. And so each individual is going to present uniquely and also need a unique approach. And so the way we figure out what approach is going to be best is to do testing to help really pinpoint that. So one size fits all. Sometimes we think of, oh, you have X condition, you need such and such an approach. That sort of one size protocol for things. And I found over the years that that's just not really effective. And that's actually how I came up with the need and why I'm so passionate about a personalized approach, because I found that that wasn't actually helping individuals as much as we would hope. So that's how I really got passionate about personalized nutrition. So the same diagnosis doesn't always have the same underlying factors. In fact, in autism, we often say you've met one person with autism and you've met one person with autism, because everybody is so different. And so labs really help us identify those root contributors. What we want to prioritize, what systems we might want to look at first, and then which targeted interventions might be the most helpful. So the four I wanted to really hone in on today that I think are some of the biggest factors for autism, ADHD, and anxiety are oxidative stress and that redox balance, methylation, and the broader concepts of one carbon metabolism, mitochondrial function, which we need for energy production, and nutrient status. And all three of these conditions have those four factors, maybe a little bit different amounts for different people, but as a whole, they're often seen in these conditions. And so these are not just isolated, these are interconnected, and they can get fairly complex. So we often have bidirectional relationships where, let's say, mitochondrial function affects oxidative stress, and then oxidative stress affects mitochondrial function. Or the same thing with methylation, that can affect redox status, and redox can affect methylation. So there's a lot of these relationships. There's also, methylation is going to affect how we produce glutathione, and that's going to affect our redox status that we'll talk about. So there's so many different pathways, and they can interrelate with each other. And then if we add on to that, these systems can be impacted to things that are going on upstream, like the health of our gut and the balance of our microbiome or dysbiosis, inflammation, different environmental exposures. And so that's why it's really helpful to get a sense of what's going on to the best that we can. So let's start with oxidative stress. So oxidative stress is really, I think that oxidative stress is at the core of really all chronic conditions. Oxidative stress creates inflammation. That's going to affect, and be influenced by the immune system. It creates neurotoxicity, and so this can damage the brain cells and disrupt our neurotransmitters. And it can contribute to mitochondrial dysfunction, which can affect our ability to produce proper energy and can increase stress on the cells. And so what we have here is the balance or the imbalance of free radicals and antioxidants. And we need this system in balance. We need some of both of these, but we need to have this flexible system that can help. We need, like, for example, we need a little inflammation from time to time, but then we need to be able to reduce that inflammation. Or we might have just natural free radicals that get produced, but we've got those antioxidants that help to balance it out. That is what we call the redox balance, reducing versus oxidizing. And so that's where we need this balance. And when we don't have it in balance, that's when we end up with oxidative stress and various challenges. And glutathione is our master antioxidant. And so this is very important, and it really helps support and protect the cells and restore that balance and help to support detoxification as well. So we'll come back. We're going to mention glutathione a number of times. So that's how that fits into all of this. So we look at some of the research. We see lower glutathione redox ratio in autism. So we have more oxidized glutathione, less reduced glutathione. So we don't have enough of that good glutathione that we need in order for all of those functions that we talked about. ADHD, we see something similar. We see oxidative stress associated with ADHD. I like to bring in studies. So I tried to bring in some studies throughout. Don't want to make it too over the top in that, but just so you see that there is science associated with what we're talking about today. And then we can also see oxidative stress is found in anxiety, depression, bipolar, and schizophrenia. So a variety of those conditions as well. And then improvements in ASD or autism spectrum disorder have been seen with methylcobalamin or B12, methyl B12, and folinic acid, a form of folate. And so we see not only do we have these lower levels of this glutathione, but the nutrients that are needed to build that glutathione, when we add those in, we see improvements in that redox status in autism when that's done. So redox imbalance can impact neurotransmitters, immune function, detoxification, and is really often a core driver across all of these conditions. And as I said, I find in the research in most chronic conditions we're looking at. So if we talk about methylation is, I think, a really important topic that is getting the press and excitement that I do think it's due these days, because it does so many things and is so important. So methylation is one part of this, what we call one carbon metabolism. So I'll explain it in a minute with a graph that I think will help a little bit. But let's just say this methylation and one carbon metabolism are really important for neurotransmitter production, as well as detoxification and DNA methylation and glutathione production. So methylation we'll talk about is the process that involves having a methyl donor attached to or activate something. And so that's important for neurotransmitter production. Detoxification, there's many processes of detoxification that involve methylation. We have to methylate our DNA, which is a epigenetic process of our gene expression. And so that's a really important one, because that also then affects our genes. So it's not only affecting systems and processes directly, but also our genes that then affect as well. And then glutathione production. So we'll talk about how all this comes together. Okay, so we often think of all of this folate cycle, methionine cycle, and transsulfuration pathway as, well, we often think of the first two as kind of methylation, but if we really want to get kind of technical about it, the methionine cycle is really what we're thinking of when we think of methylation. That's where SAM, or S-adenosine methionine, produces this methyl donor that then, as I mentioned, activates all of these things. It's an important part of the process. And so it's important for these, methylation reactions and neurotransmitter metabolism. But along with that, we need the folate cycle, which feeds into the methionine cycle and makes sure that homocysteine gets recycled back up to methionine so we can go around this cycle again. So we need this folate cycle because we need 5-methyl tetrahydrofolate, that specific form. We need that to get in here along with B12 to make sure that this happens. So that's why these two you'll often see hand in hand. And then we also then need this methionine cycle, this homocysteine, to go down. If I were gonna if I think of this, and I kind of, if I actually put the homocysteine a little bit lower and I kind of put SAM and SAH over here, put homocysteine here, then you can kind of think it goes it'll go down. And so homocysteine goes down to the transsulfuration pathway. And that's where we get cysteine, thionine, cysteine, and then glutathione. That's how all of these come together. So I wanted to talk first about glutathione because, and oxidative stress specifically, because I think oxidative stress is so central to all conditions. I think it's good to start with that one. But we kind of need to back up a little bit to the methionine cycle, which actually comes before the production of glutathione. So, in order to have good glutathione and oxidative or redox status, we need good methylation and we need a good folate cycle working. And these systems, these pathways, they are all interconnected and they need a variety of nutrients. So I mentioned folate and B12 already. We also need B6 as part of this process. We need riboflavin and niacin. We also need zinc and magnesium. So later we're going to talk about some of the nutrients and why they're so important in this conversation. It's because they really feed and fuel these pathways, at least as part of it. I mean, they do so much more than that, but in relationship to this particularly. So that's where I think we hear so much about methylation lately because it's so important. If we look at what is the neurotransmitter connection, I'm going to call it one carbon metabolism because that's technically more accurate. Why is one carbon metabolism so important for the brain and these conditions that we're talking about? Well, we need that SAM in order to go ahead and have proper COMT or catecholamine O methyltransferase. And we need that to then help make sure that our dopamine is metabolized properly and that we have the proper regulation, breakdown of neurotransmitters, have the right balance of neurotransmitters. And then for BH4, this is very folate cycle dependent as well as redox dependent. And this is going to make sure that we have proper serotonin and dopamine production. And so you can see here, if we look at one carbon metabolism, these are just a couple of the ways that they are really important to the making sure that we have the brain chemicals that we need for our mood, for our behavior, for attention. And so I think that's why, that's why we hear about it a lot because it's important. And we also see that often the nutrients needed can be deficient or not sufficient. And so, and we won't go into all this, but there are certain like gene SNPs and things that we do know for things like autism that are influenced, which also help to explain a little bit of this, but we'll leave that for now. So we look at some of that research, we see a decreased SAM to SAW ratio in autism, and we see deficiencies, B6, B9 or folate, and B12 deficiencies in ASD, in autism. And so when we see these deficiencies affect methylation. And then, this can lead to elevated homocysteine, reduced levels of glutathione. And I guess I shouldn't say reduced glutathione because we kind of want reduced versus oxidized glutathione. So what I mean here is reduced levels of glutathione. And then functional folate deficiency. And so those are some of the ways that those are involved in what the research is showing. Now, histamine is an important piece of this because histamine is a good clinical marker. So histamine has to be broken down by methylation. So there's an enzyme called HNMT, which is histamine N methyltransferase. And this is needed in order to break down histamine. So it's a helpful marker for methylation. So when histamine is high, we're not getting that proper breakdown. We have reduced histamine breakdown, and that can reflect lower methylation status or capacity. When histamine is low, that may reflect higher methylation or what some practitioners call over-methylation. So this histamine marker can be very, very helpful. Mitochondrial function. So mitochondria are the energy, the way we produce energy in every cell. The brain has a very high demand for energy. And so mitochondria, when they're not functioning well, can really have a dramatic effect on the brain. And so that can affect cognition and mood and also the whole entire body because every cell in the body and every organ system requires this energy. And so when we don't have good mitochondrial function, we can have system-wide effects. This is a very interconnected system. So when you look at methylation and transsulfuration, they support the mitochondria. So they provide key nutrients and antioxidants and energy that help or that support that energy to help with this mitochondrial function. We also when oxidative stress damages mitochondria. So when oxidative stress is high, this can impact mitochondrial function, have more free radicals. And then as I mentioned earlier, unfortunately, those increased free radicals can damage the mitochondria. So that's an important piece of it. And so it becomes this self-reinforcing cycle that when mitochondrial dysfunction increases, this can further impact methylation and transsulfuration in a negative way, and that can perpetuate the cycle. So supporting these systems can also help protect and support the mitochondria. And so that's a key piece of this. We look at the research for that. There's quite a bit in autism. Mitochondrial dysfunction is quite common. It depends what study, anywhere from 30 to 50% of individuals, up to 80% in some populations of autism may have mitochondrial dysfunction. So it's, we often think of, well, we don't necessarily, but in the medical community, they often think of mitochondrial disease. I haven't seen quite as much focus on just dysfunctioning mitochondria that are not functioning optimally. And that's what we're talking about here, is that the mitochondria are not functioning optimally. And so I think this can sometimes get overlooked. So this one also is not always measured directly. So we're often looking for organic acid markers, maybe lactate, carnitine, CoQ10, or other related oxidative stress markers to get an assessment of how the mitochondria are doing. So let's talk about those nutrient-dependent processes. So as I had mentioned, nutrients fuel all of the things that we've just talked about, and obviously so much more, right? We need nutrients to make sure our gut is healthy and working well and continually repaired as needed, and to build neurotransmitters and to build digestive enzymes, everything. We need nutrients for all systems in our body. As well as the specific ones we talked about today. So nutrients really are important for influencing our neurotransmitters, our energy, our antioxidant capacity. So let's talk about the three, folate, B12, and B6. So these support methylation and transsulfuration, as we just talked about. B6 is actually directly supports neurotransmitter production. And so this one is particularly important as well. Deficiencies have been linked to autism and ADHD as well as anxiety. And then supplementation has been found to be beneficial. There was a study in autism that I wanted to highlight here. And that was it was a study that looked at a variety of different nutrients for individuals. But we also see in practice a lot that these supplements can be very helpful with ADHD and anxiety as well. And there's a correlation of higher intake of these nutrients with reduced odds of having anxiety, depression, and psychological stress. Another set of nutrients is zinc copper, and the importance of this zinc copper balance. We need these nutrients, but we do need them in balance. And zinc is particularly important and seems to get out of balance a little more easily. It's important for GABA, so that calming effect versus glutamate, which is more of that excitatory. We need that support from zinc, as well as a variety of other factors and ways that zinc is really important for antioxidants and all sorts of things. And again, we need both of these, but we need them in some sort of balance. So when they're not in balance, and we have low zinc and high copper, we have increased oxidative stress very often. We get imbalances in neurotransmitters, and then we get symptoms, behavioral symptoms, and things that can come from this low zinc and high copper. So zinc and copper ratio is actually a really informative way of understanding these nutrients. Rather than just these markers in isolation, also what is the ratio of these is really, really helpful. And they can impair really key systems. There's a study on autism that looked at zinc and copper and found that they were very important with antioxidant status. Metallothionein, which is an important process of detoxifying heavy metals, GABA and glutamate, influencing autism symptom severity and neurological function. So they found that this zinc copper status was very important. And then another study showed that decreased zinc copper, so lower zinc, higher copper ratio, correlated with the severity of autism. So really important nutrients to understand. Then cryptopyrrole, or also what's known as HPL, this is a marker that is very helpful to understand because cryptopyrrole binds to zinc and B6 and can deplete them. And we've talked about how important zinc is and how important B6 is in all of these things that we've talked about. So they can impact the neurotransmitters, they can impact oxidative stress, and they are important in they've been found in ADHD, anxiety, and autism. So these markers are important. HPL, I wrote it down because I can never remember it. Hydrohemopyrrolin 2,1. But most of us know it as cryptopyrrole. And then magnesium is important for all these different systems. It helps with these pathways, and it's also important for mitochondrial function, redox balance, methylation, and neurotransmitter regulation. So magnesium is a really important nutrient as well. Some of the research on magnesium show lower levels of magnesium in ADHD and autism. There also there might be a bit more anxiety and stress. So lower levels of magnesium are found in ADHD and autism, and that's these two studies here. And then this third study shows that magnesium supplementation may help reduce anxiety and stress. And what we see often in practice is a calming with magnesium. Magnesium is very calming to the nerves. It just provides that calming effect. It can help with constipation as well as, and it's a very helpful nutrient. All right, so let's talk about this in clinical practice here. So let's talk about labs and how we can use labs to help identify these drivers as well as to prioritize what systems to look at and what interventions and personalized nutrition would be most beneficial. So I want to talk about a few categories of labs here. So oxidative stress, methylation, mineral balance, cryptopyrrole, and additional markers. So oxidative stress markers, I often like to think of glutathione. And so there's reduced glutathione, which I'll call the good glutathione, and then oxidized glutathione, total glutathione, and then the glutathione redox ratio of the reduced to the oxidized. And understanding this picture of glutathione really helps to get a sense of oxidative stress. We look at methylation markers. We look at whole blood histamine. Remember we talked about histamine earlier. So whole blood histamine can be very helpful, and an elevation is associated with undermethylation, while a low histamine may suggest overmethylation. Look at serum B12. That can reflect the circulating levels of B12, but that's often nice in conjunction with MMA or methylmalonic acid, which is a bit more of a functional B12 marker at the cellular level. We can look at serum folate as well as red blood cell folate, whether we're looking at the short-term dietary folate status in the serum or more of the longer term storage in the red blood cells. Also, there's homocysteine. So remember, there's that process, and in that methionine cycle, there's homocysteine. This marker reflects impaired remethylation or transsulfuration, and so that it can indicate either an insufficiency of B12 and folate B12 and folate to remethylate it, or B6, which takes it down to transsulfuration. And then the other is SAM and SAW and the SAM-SAW ratio. That's another direct marker of methylation status, since SAM is the methyl donor, is a universal methyl donor. And then for minerals, we want to look at plasma zinc, serum copper, ceruloplasmin, and then the zinc copper ratio. These are very helpful to assess that zinc copper status. And then looking at cryptopyrroles, so looking at that urinary marker of HPL and that indication of zinc and B6 deficiency. There's also additional markers that I think can be helpful. So looking at amino acids, iron or ferritin status, magnesium, often red blood cell magnesium, omega-3s can be another one. Organic acids can provide some good helpful information, and then gut microbiome testing. So if we look again at that, what's upstream, is the gut and the microbiome, could it be depleting some of these nutrients? So pathogens in the gut can give off certain compounds that can deplete some of these important antioxidants and things. And so sometimes we want to look beyond and kind of get a bigger picture as well. So there's a DHA metabolic panel plus that has a lot of these great markers to get started. So it's a panel that has what I think are 5 of the key markers that really help to understand that brain chemistry and that free copper oxidative stress, to really be, just an efficient starting point to get a sense of what is the nutrient status, what does that methylation status look like, what's oxidative stress looking like, what's that serum, what's that zinc copper ratio. And so this panel consists of the cryptopyrrole urine test, plasma zinc, serum copper, as well as ceruloplasmin, which is the copper binding and whether there's free copper. So it gives you a bit of that bigger picture of what's happening with zinc copper ratio, as well as whether that copper is free and what's happening there. And then whole blood histamine. So this panel really puts together a lot of those key pieces that we were talking about, and can really be, I think, a really effective starting point to identify some of the common underlying imbalances in autism, ADHD, and anxiety. And these are often some of the key markers that practitioners in this space often will look at. So DHA is offering a webinar special 30% off. So if you can use the promo code Julie Matthews webinar special to get that 30% off. So check that out. And just if we putting it all together before we end here, we can start by identifying the biochemical patterns that are most relevant, and then matching that with the systems and the deficiencies, and then putting together the interventions, usually often layering them as appropriate. Can't do everything all at once, so we want to usually kind of layer the different strategies based on what we find and what makes the most sense. And so when we use that type of targeted, precision, personalized nutrition, that may really help support and improve behavior and mood and focus and energy and overall neurological function. So, key takeaways for today are that there's shared biochemical patterns that underlie many conditions, particularly neurological conditions and neurodevelopmental conditions. And the systems are interconnected as well. And many of these systems are even bidirectional, really being heavily influencing each other and having this kind of complex interplay. And nutrients drive these systems, and so deficiencies are important to uncover. And labs can help provide a really good clarity for how to implement a personalized approach. And then that personalized approach can lead to better outcomes with the individuals that you're working with. And, there's a lot going on with clients and patients, and when they can feel confident that recommendations are based on something that they're looking at, something that's concrete. I really think it helps with compliance. It also helps, most importantly, with results, and rather than kind of having to do more troubleshooting and guesswork, you can get some good clarity. So personalized lab guided nutrition really helps bridge the science and the clinical application and the results that we get with individuals, that those real world outcomes that are so important. So with that, let's go ahead and get to questions and answers.
Hey, everybody. I'm not sure if Dan and Gajen are going to join us live. Hello.
Hey, I'll jump on, Julie. I'll run through some of these questions for you. Maybe we'll
Great.
take them from top to bottom. What a great presentation. I appreciate that. It was really, really lovely. Um,
Thank you.
well, hey, starting out, curious, the homocysteine levels you are seeing with the methylation challenges. So what levels are you seeing in homocysteine results with methylation challenges?
What's interesting is that we often think of high homocysteine in a lot of people, but with autism, it often is low homocysteine or even just normal homocysteine. So high homocysteine is typically associated with um, problems with remethylating or challenges with going down and doing transsulfuration. But with people with autism, they often have poor methylation, so just kind of not moving around that methionine cycle very quickly. So it might even look low or normal. So it's not always high like you might see with people with heart disease or something like that. So it does depend. I would say if anything, I might see it low, but it does really depend on kind of like what's behind it for the person.
Right. So to piggyback off that, I'm asking you this question. It's not coming from the chat. Is that why you're measuring that with those additional markers? You're really using that whole, slew of tests that I saw that you put up on the screen. If I recall correctly, it was not only the homocysteine, but you were looking at whole blood histamine, like B12, MMA, folate, some SAM-SAW stuff. Are you looking at all that up front? Like, do you sometimes just see a piece of them? Do they all play together? Speak a little bit to that.
Yeah, it does. Yeah, the reason for that is the bigger picture. So, you know, uh, some people might just start with one, they might start with whole blood histamine, but if they're having trouble getting the big picture, they might pull in some of the others. Sam Saw is a nice one because it's a direct relationship of the methyl donors of SAM to the SAW, which is the step after. So that's kind of a nice one if they want to clarify it more. And then B12 and folate are helping us figure out if we have that folate cycle that's going to feed into it, and maybe a bit of the reason of why some of that isn't working as well.
Sure, absolutely. You get a good cycle with that report. It shows a lot.
Mhm.
What about kids with AFID? How do we get nutrients into them, especially with allergies to ingredients and most supplements?
Yes. Uh, that's ARFID. ARFID is it ARFID? Is that what they want?
I'm reading out of the
Tell,
chat. I see an AFID. However, it could be
AF.
close to an R. And Danielle, if you
I'm
want to
thinking
write
of
in more.
ARFID, which
Yes,
is
she wrote
yeah, I'm
yes.
guessing I'm guessing that's what that is because it's real but ARFID is basically a feeding disorder, kind of like a bit in between like picky eating and it's more of a significant feeding disorder than something like picky eating would be. And so if it's picky eating, it's probably a little bit easier for me to answer because ARFID might really require some psychological support and some particular interventions around it because it is more of a kind of a specific diagnosis than the standard um, picky eating. So picky eating is very common in autism. About 80% of kids with autism have picky eating and very specific picky eating habits. But kids with autism also have higher levels of ARFID than other conditions as well. I think of them kind of differently. So if we're talking kind of regular picky eating, I look at things like texture. That's a really big issue. And that's something that usually we can use to support um, crispy and crunchy foods, things like that. Sensory issues is one of the main reasons for picky eating in kids with autism and other types of developmental conditions. So if I were going to be approaching from that side, I'd try to get creative with what textures they like, what flavors they like. Do they like things separate or together? How can we make vegetables crispy, crunchy, things like that to get more nutrients? Some of these important B vitamins and other nutrients in. If it's ARFID, I mean, these might be tools, but I do think that since it's more of a clinical diagnosis, it might need some further support.
Sure. I promise I didn't plant this one, Julie. It's from Alan Kadish. But what lab are you using for your HPL evaluations?
Great question. DHA is really I think the preliminary, lab for cryptopyrrole for HPL. They've been doing it. I mean, Dan can speak better than I can, but they've been doing
Sure.
it forever and feel like it's one of the most accurate. But what I'm going to actually turn this one over to you because you will know even
Yeah.
more history than I do.
Yeah, happy to discuss it. So the molecule itself, the hydroxyhemopyrrolin, the 2-1, as you mentioned, is a light temperature and time sensitive molecule. And so it's also a test that's not regulated by the FDA. You really have to trust the laboratory that does it and know they're following a process that respects those sensitivities of the specimen. So truthfully, it's all about the kitting process, the receiving process, and the testing process in the laboratory that makes it a great test. And we did lab develop that test. We service, other laboratories and providers in 22 countries because it's such a hard test to develop and maintain. But ultimately, what makes it really special here, it's our ability to ensure that we're shipping overnight and receiving them first thing in the morning, breaking them down as soon as they're in the lab and putting them directly into the test run without any exposure to light and letting them get warm and things of that nature. Running a really strong procedure and also correcting it for specific gravity, more or less the concentration or dilution. So you get the highest quality test here from those factors.
Great.
Cool.
All right.
Yeah, let's jump on to the next one. What are your recommendations of zinc and B6? Excuse me, what are your recommendations when zinc and B6 are normal or high and cryptopyrrole is high as well? Would you still supplement? Thank you.
I think it does depend, and this is similar to that other question about just understanding the bigger picture. So, what else is going on? What does the zinc level look like? What does the zinc copper ratio look like? What does the ceruloplasmin look like? What symptoms do they have? Are they currently supplementing? Because there can be normal levels, but functionally it's not working well. It's not getting into the cell, it's not being transported, or whatever that might be. So that's, I think, kind of a key, kind of a key factor is to just understand the bigger picture. Why is that? Why is that HPL level high? Could it be like oxidative stress or something else that's a factor? So that's why I feel like that's what's nice about a panel. You're getting a number of things to give you a little bit of a bigger picture when you want to make that
Certainly.
choice.
I think you touched on a key point there, something we see in the lab a lot, and we ask about them on the intake forms from patients who are completing the tests, right? Like, have you
Mhm.
supplemented in the last 12 to 24 hours? And sometimes you see that a yes, even though the recommendations are not to do that, right? Or
Right.
sometimes people are taking sports drinks or liquid IVs and things, and we don't recognize
Mhm.
that those are also supplements. So, some important factors that could be at play there from the lab standpoint. Julie, for magnesium, which lab test would best reflect the level?
Well, usually it's RBC, red blood cell. That's the one I usually think of.
Absolutely. We see that a lot too. Um, how do you know if a high B12 is due to the body not being able to use versus some other reason? Is an
Mm,
HTMA test reliable in this
mm, mm.
circumstance?
That's a good one because sometimes B12 is quite high, but it's just not being utilized. I personally like MMA, methylmalonic acid, for that to get more of a functional picture of whether B12 is working well. Uh, so I personally use that more than hair testing. Hair testing can be accurate, maybe some minerals more than others. I just tend to like, especially if we're looking at B12, I really like to look at MMA.
Certainly. Okay, what about vitamin C deficiency and how
Mm.
that impacts the gut-brain axis, lymphatic health drainage? Do you feel that this is also a big contributing factor that is seen heavily in autism and presenting leaky gut, chronic gastritis?
This is such a great question because I teach practitioners in my bio-individual nutrition training, and just this Tuesday, so 2 days ago, we had this very conversation about vitamin C. And what's interesting is vitamin C doesn't get talked about a lot, and that's actually one of the things we were saying. I think we get very much excited about like the latest and greatest kind of exciting new nutrient to come out, and we kind of forget some of the basics. Like vitamin C has been touted for so many decades, it kind of, I feel like, gets ignored. So I absolutely think it's very important. Vitamin C is, a major antioxidant. It's also one of the more common deficiencies in autism, especially in picky eaters. So I do think that it's key. What's interesting is you hardly hear about it. It's not one that I find gets studied as much or gets touted as much, but I absolutely think it's an important one.
Sure. What form of magnesium do you recommend for anxiety and stress?
I mean, for anxiety and stress, I would say two I like magnesium threonate and magnesium glycinate. So I think of magnesium glycinate a little bit more for like physical calming stress, and threonate a little bit for mental calming stress. Those are two of my favorites. I mean, I might look at citrate or something else for other needs that people have, but if someone's talking more about that calming kind of stress sort of thing, those are usually my two favorites.
Sure. Joy Gentle would like to know, does it ever make sense to measure RBC zinc and RBC copper?
You know, usually the gold standard is the plasma and the serum. And there may be other reasons, I suppose, that some people might want to know, other factors, storage versus this versus that. But typically, when we think of it, we think of plasma zinc and serum copper as the two kind of, and then because we can do the ratios on those as well.
Sure, absolutely. Yeah. Most studied, right? We can report the optimal functional ranges. We're using those key
Mhm.
tubes that really create like great stability.
Mhm.
So, absolutely. Okay, what does low homocysteine indicate in the body and what causes it?
Mm. Low homocysteine typically is low methylation, typically. Now, it could be also well, I'll get to it. It also could be a like a sulfur issue in that it's going down to glutathione. The needs for glutathione are high, or there's some issue and it's all funneling down to glutathione rather than recycling and staying up appropriately in the upper part. But typically, if we see low homocysteine, it's because there isn't an adequate methylation. So maybe there's not the proper amount of folate and B12 bringing those methyl donors in and moving them, or the methyl group and moving it around to make that SAM create that methyl donor there, or provide the methyl donor. Typically, that's what we see. Um, if it gets stuck, sometimes it might get high, but if it's low, I usually think that that's kind of just a slow process, or it's just kind of like it's going out the hole in the bucket down to transulfuration.
Certainly. And this is me again. I love that that's the answer, right? Because we hear so many people come to the laboratory and they're running MTHFR and making the assumption at under methylation, right? And throwing methylated folated people. And what I noticed about your methylation markers is we aren't using genetic markers. We're looking at the opportunity for over methylation as well as under methylation. And it's very smart stuff. So I appreciate that answer. What are the zinc to copper ratio that you're looking for with in general, it sounds like, as I read the question.
Yeah, I mean, usually I think of it sort of like a like a like a 1 to 1 kind, like, I don't know, 0.9 to 1.3. If it's too low, which is most common, then that's going to be low zinc compared to copper, and that's where we get that um not enough zinc, which is much more common than if that ratio goes too low, then we're getting low zinc, high copper. And then if it goes too high, I suppose we get high copper, low zinc, but it's pretty rare that, my colleagues and I really talk about seeing zinc go too high and copper go too low.
Sure.
So copper, they compete for absorption as well. So you do want to be a little aware, and that's why testing is really good, because we don't want to just throw tons of zinc at something. You do, copper often gets is like the evil villain, but we do need copper. We need copper for the mitochondria. We need copper for all sorts of things. Now, typically, most of us get enough copper. A lot of people don't need to supplement any extra copper. But I do like to say, sometimes there's like always too much of a good thing. If you supplement too much with zinc, we could theoretically not have enough copper. But typically, I see the opposite. I typically see that people have a supplement that has copper in it and they are depleting their zinc more. That's a much more common scenario that I see. So a lot of like autism formulas don't even have copper in them, because most people with autism are fine with copper and too low in zinc, and we don't want to drive that zinc lower by adding more copper typically.
Sure. Absolutely. Okay, from Danae McKitten. Having a dental background, have you assessed nutrient deficiencies in the mouth for those adverse to blood testing?
Hmm. Nutrient deficiencies in the mouth. I don't know how she does it. Does she do it based on like symptoms like bleeding gums, or is she doing it based on like a saliva test? I don't know. I don't know anything about that. Maybe
Yeah.
as a dental expert, she can share more.
Yeah, Danae, write us in. Always good to hear from you. It's been a long time. Shoot me an email and I'll loop in Julie and we'll chat. From Marta Long, she thanks you for your wonderful presentation and says, Assuming no lab error, including incorrect collection, what would be some reasons for low cryptopyrrole in severe autism spectrum disorder when the copper is severely elevated in relation to zinc? Would you empirically treat or repeat testing later or accept that value? Are pyrroles
Mm.
affected by antipsychotics? So we've got
Oh,
a pretty layered question there. Let me know if you
yeah.
want me to repeat or dive in.
No, it's a good question. I don't know the psychiatric, meds question. That one I don't know the answer to. I haven't looked at it from that perspective. Low HPL with low zinc and high copper. I usually think of, what other reasons for high HPL other than that like zinc B6 thing for low. I don't know. That's a good question. I don't know if does your lab have that reference range and have any thoughts? I don't actually have a good answer to this question. I don't know if your lab has come across anything or any of the practitioners you work with have any good thoughts on that one.
No, I would reread the question. It jumped out of the chat when we answered it. I might have to circle back on that one and hit it by email, but good thought. What came to mind for me is just in general, right? You've got low zinc, you've got super high copper, we've got optimal pyrroles. That's not uncommon, right? I think there are people who don't have pyrroles at the root cause of a zinc deficiency, and there are
Yeah.
people who have environmental factors that cause high copper. And we do see that in the laboratory from time to time. It was a fairly layered question, though, so I don't want to
Yeah.
misinterpret the request. So over time, feel free to shoot it in via email, and we'll jump on it. But from Katie Williams, what do you suggest for someone who gets nauseous taking B vitamins?
Oh, whew. I mean, take it with food. It would be the top one. If they tend to be nauseous, some clients tend to be more nauseous in the morning, where their meal in the morning isn't as big, and therefore that's a factor. So I like to put it with a pretty substantial meal. I mean, usually B vitamins you're going to want to take at least twice a day because they're water soluble. But if they can't get them in the morning, maybe they can do like lunch and dinner or something like that. I find it's tricky because also some B vitamins might be too stimulating at dinner. So I think here's what I would probably do if it was my client. I'd try to figure out which nutrients they really need and which ones are creating the nausea and see if I could either get the amount in a low enough amount divided, like maybe they could do a smaller amount 3 times a day, or maybe what they really need is I mean, B6 is one of those ones that can be a little stimulating, but then also it can be a little nauseating. So that's where it can get a little tricky. You could try the different forms. Sometimes form makes a difference. Uh, so I think those are the kinds of things I would look at. Sometimes people do better with individual B vitamins. I mean, I know they all work in concert, and ideally we take a B complex, but it's not uncommon for me to do individual Bs with people because then I can pick the time of the day a little bit better for certain ones, and I can pick forms and I can pick amounts. Some people need a lot of B6 and they do just fine with it. Some people just can't handle like thiamine or something like that. And so if they try to take a B vitamin, they don't get any in because they can't tolerate the B complex. Whereas if I just do B6 by itself or B6 and B12 and folate, then those tend to be tolerated. So that's often the strategy I will do, honestly, is to do individual Bs that I think they need, or smaller levels of a complex.
Sure, absolutely. Okay, here's a good one. For those that are on ADHD medications and experience appetite suppression from the medications and struggle with eating and taking supplements, do you have any recommendations on how to best manage nutrition and supplements better?
I'm going to try to approach this more from the, person that is struggling maybe with appetite rather than any other effects. Since I'm not since I'm not a doctor, I don't do medications. I'm going to think about it more from like, oh, how would I help somebody that was having these challenges? I look at, is there a time of the day that they have more of an appetite, kind of similar to that other one, but from a little bit of a different angle. So, again, do they have more appetite as the day goes on? Can they do their supplements then? Again, it's nice to often get nutrients twice a day, but sometimes we just have to do the best we can and get them in when we can. So I would say, number one, for foods, try to make those foods as, I want to say, as delicious as possible. And I know that seems really basic, but like, again, can we get more vegetables by doing like kale chips where we get some good folate in? Can we add a little natural, like a little salt? Like we're not talking like fast food 3 times a day. I'm just talking about well-seasoned foods that might be more appealing to the person. Forms they like, things they like. So from a food perspective, and then from a supplement perspective, I'd go back to, is there a particular time of the day that they do better? Or if, like that other person, you have to get supplements in, but it's difficult because maybe they're nauseous. Can we get some good food that they like that's also healthy in with that food, and try to, it's interesting. Sometimes people will be, whatever, maybe anxious in the morning and they can't get a lot of food down. They can't eat too many really heavy foods. Maybe they do better with like yogurt or non-dairy yogurt and some fruit. Other people, really depends on the person. But I would look for foods that maybe it's in like a smoothie form where they can drink it, that kind of thing. And maybe there's you can get some good nutrients in that way, or enough nutrition that you can get a little bit of supplements. And then, I would just, again, just try to get to the bottom of it from that personal perspective of what might be the biggest challenge, and try to work around it a little.
Definitely. Okay, let's grab a couple more. I know we're going over. I appreciate your time here and your flexibility and your attention to everybody's questions. We've got a couple good ones and we've got a big long list. I'm going to run through some great ones with you, and then I think we'll call it to an end.
Okay.
Here's a good one from Rachel. For clinicians without experience on nutraceutical management, how do you recommend they start learning age-appropriate and symptom-appropriate dosages?
Age appropriate and symptom appropriate dosages. Well, the symptom appropriate is, there's a really, I think, a very cool app. My mentor, Dr. James Adams, we wrote a couple papers together, and one of the adjuncts, so we did the paper on diet, but he also did a paper on nutraceuticals, and he looked at dozens of different nutraceuticals used in autism and the symptoms that they helped with. And so you can either look at the paper, which is, I think, the ratings of the effectiveness of nutraceuticals or something like that, or there's an app which is Autism. It's it's Autism Nutrition Research Center, or Autism NRC. I forget exactly. It might be ANRC Rater. One of those things. And it's cool because you can go into all the different symptoms and see the top nutrients in autism, the top nutrients to help a whole variety of symptoms. And it goes into diets and it goes into medications. It's very extensive and really very interesting. So from a symptom perspective, I think that might be a great way to start to see what families say, because there are hundreds of people, if not thousands of people, depending on the portion of that study that were questioned. And then the second part was, dosages. Usually dosages are often based on weight. So, things like RDA, there's great information on vitamins and mineral dosing there's RDA, but then there's also optimal levels. And so that is a little bit different. Where would I say for optimal levels? That's a little trickier. You might have to find like a good practitioner book that you like that's got some good levels for more optimal levels for things. They're out there. But yeah, RDAs, now that I think about it, might not be sufficient for like the types of things that we're thinking about. My colleagues wrote a good book. Dana Lake, Laake, she and Pam Compart wrote a book on autism and ADHD nutrients, and they have much more of those kind of optimal levels for things.
Sure. Another, great option. I've seen a number of providers do this too, is like contact a great compounding pharmacy, right, who has clinical staff that can help you. Village Green Apothecary is a great one that comes to mind that I think about. So another good option there, right?
That's a great idea. Absolutely. Yeah. And the nice thing about that is, and then when they put that formula together, and this goes back to the earlier question about that person trying to get the B vitamins or the this or that, you can really customize that formula to the forms that you like, the amounts that you like. So if there's certain things that are causing a problem, you can, instead of, my way was 27 different individual nutrients in the way I just described it. But it's
Sure.
like I should have, thought of that, because that's a great way to do it. And is to then you can dial in very specifically the dosages, the forms, and then it's much more manageable than trying to do a bunch of different things.
Certainly. Okay, let's get a couple more in. Do glutathione patches or other vitamin patches work as well as oral supplements in your experience?
It's funny because literally at that same call that I mentioned where we talked about vitamin C, someone mentioned glutathione patches. And in fact, I could probably go through my stack of papers on my desk right now and figure out which one they were referring to. One of my colleagues on the call was saying that they really do like glutathione patches and found them to be very helpful. So for whatever that's worth, I haven't used them too much. So I don't have as good of info, but the nice thing is that, well, glutathione can be it's very sulfury. So it's hard to do. So that's where the patches can be particularly helpful. And was there any more to that question that I missed?
No.
Okay.
Okay, let's hit two more. When do you recheck labs after starting supplementation? 2 to 3 months?
I would say a little, maybe a little bit longer. I might say it depends. Again, depends how long to get somebody on board. But once they're rolling along, I'd say 3 months, 3 months, 3 to 6 months. It depends. I mean, something like fatty acids might take longer, but if you're talking about vitamins and minerals, probably 3 months is probably pretty good. It also does depend on, you know, everybody's got a certain budget, so it depends on when you want answers and how, do they have the ability to do it every 3 months? If they don't, then, kind of just figuring out what's going to be the best for putting their plan together for them.
Sure. Okay, I lied. I'd like to take two more. There are two great ones in here.
Okay.
Talk more about measuring histamine levels in blood. I do not typically test histamine because it has such a short half-life. It only lasts 30 to 90 minutes in the bloodstream. What is the usefulness of testing histamine?
Yeah, well, whole blood histamine, because it's broken down by methylation, it can be used as a marker. So if it's higher, then we can, get a sense that there's not enough methylation to be breaking down that histamine. If it's low, some people think, okay, well, maybe there's some over methylation happening and it's going on the lower side. That's usually why it's definitely a marker that a lot of practitioners do use for methylation. It's been used for a long time now as a marker for methylation. And I think it's a great starting point. And then I think if people are, everybody is pretty complex in terms of there could be other things reasons that for maybe them, that histamine marker may not be the best marker. And that at that point, if you're not getting, for example, if I had somebody, which I've had before, where the histamine level didn't quite equate to the symptoms that I was seeing, then I might look a little bit more at some of those other markers of things. But sometimes, it can be a good starting point, especially if you're doing something like that panel where you're getting, 5 or 6 things all at once. If it's painting that picture that makes sense clinically, I feel like, and I've heard this from, Walsh and Mensa and other, really amazing doctors and practitioners and researchers, that clinical picture is a really important piece to go with the lab testing. So if that clinical piece is matching what you're seeing, then you're probably in pretty good shape. If it's not, that's when you can look at other tests that might give you more of the picture.
Certainly. Okay, let's do one more here. A common question I hear parents ask is, how do I get my kids to eat different foods? How do you motivate a child to eat, these different quality foods when their food preferences are limited?
Yes. That's very common. As I said earlier, about 80% of kids with developmental delays have picky eating. And kids in general just tend to like, carb foods, white foods, sweet foods. That's just sort of built into our, nature, I suppose, unfortunately, to some extent. So I like to look at, like I said, I guess my big thing there is what forms do they like? Do they like when things are individual, like a meat separated from a vegetable, separated from a thing, do they like crunchy? Do they like raw carrots or maybe even raw green beans? Or, they did like things like that. Are we talking really limited eaters where they're used to eating like chicken nuggets and French fries or chicken nuggets and potato chips? Do we need to start with like some sweet potato chips and some carrot chips, or just figuring out where they are in their process and what's going to help move them forward and kind of maybe make a shift in their mind. If they're eating like all white foods, if they eat like a sweet potato chip and now they're like, oh, this is really delicious, it's not white, it's orange, they might start to go, oh, okay, what, I'm going to try some of these foods in other ways too. So usually I try to take small steps, incorporating some factors that they already like. Like if they like meatballs, it depends how picky they are. So if they like meatballs, can you puree some squash into those meatballs or something else into a cauliflower into the meatballs? Or if you do that, are you going to lose that trust and now they're not going to eat that food anymore? So you have to know a little bit of the person you're working with. Um, I mean, one of my favorite things to put in like smoothies is frozen cauliflower rice. It has very little flavor. It's got great nutrients in it. Kids wouldn't even know it was in there. So I think there's a lot of creative ways in it, and it all depends on like where are you starting, what foods do you have to start with, and are you able to you know, I hate to say hi, but like some kids are like the anxiety about adding a new food is the issue, but they don't really have any problem if it's in there. They just don't know it's in there. Others, you lose their trust if you add it to the food. In that case, you would not want to add it to the food. In that case, you might just want to say, hey, we're trying carrot chips today. We're trying kale chips today. This is what we're doing, and just kind of put it out there to them. So I think knowing a bit of their disposition and what's going on behind their food restrictions can help people figure out which way is good. Also, do they eat meat? What don't they like? Some kids don't like meat because it's too chewy or it's too dry, so they don't like chicken breast, and that's why they like things like chicken nuggets. So could you make it steak can be hard to chew, so maybe if you made like a meatball and you added you can make it softer. So understanding like what textures they like as well, even in things like meat can help.
Absolutely. I'm glad we ended on that note. That was a great explanation. I'm taking some of those home personally for myself,
Great.
because all the kids are picky eaters at some level. You might get a great one that just eats everything, but it's rare, right?
Yes,
So,
exactly.
Julia, it was a wonderful presentation today. We had so many attendees. We hit the limit. I think we actually have to send out the recording to people that couldn't get in because we had more people than we'd ever had attend a webinar. What great content. Thank you so much for taking the extra time. Um, we appreciate you greatly. Wonderful presentation. Would you like to leave us with any parting words?
Oh, wow. Well, thanks, Dan. It was really a pleasure to do this and to talk with everybody. I would just say, whether you're a parent out there or you're a practitioner, I really feel, my passion is nourishing hope, just nourishing hope that no matter where people are, no matter where they're starting, there are things that they can do. There's new information that they can find that they can start to take action on, or little steps they can take once they find that information. So I'd say no matter where you're starting, there are places that you can go from there. So, keep nourishing hope.
Cool. Well, to all the attendees out there, thank you so much for coming. Julie, thank you so much for putting on a great presentation. It's been a pleasure today. We look forward to being here for everybody that attended in the future. Feel free to give us a call, reach out by email. We're always here to help and have a lovely day. Bye, guys.
Bye.